Detecting cancer with bacteria, easy, in the near future. In short, a group of scientists from the USA and Australia has developed bacteria that can help detect cancer. In a new study, the team demonstrated that these bacterial biosensors are capable of detecting mutated DNA released from tumors, which then enters the intestine due to colorectal tumors. Technical part: the bacteria are programmed to seek out and capture mutated DNA in the CRAS gene, which is associated with many cases of cancer. When the bacteria detect mutated DNA, they emit a signal indicating the presence of cancer. These bacterial biosensors can also be used to diagnose specific types of cancer that they detect. Additionally, there is potential for using these bacteria to treat tumors in the intestine. However, much work still needs to be done before this approach can be used in humans. The new technology is also unlikely to replace colonoscopy in the near future, as colonoscopy remains the best way to detect and remove precancerous growths in the intestine. To create the biosensors, scientists utilized the natural abilities of bacteria to absorb DNA from their environment and integrate it into their genomes. They used Acinetobacter bailey bacteria, which are particularly good at assimilating DNA from their surroundings. The bacteria were programmed to seek out and absorb mutations in the CRAS gene. In experiments with laboratory samples of tumors and in mice, the team demonstrated that these bacteria could detect mutations in the CRAS gene associated with colorectal cancer. However, much work still needs to be done before this approach can be applied in practice. Researchers also note that this approach could have a wide range of clinical applications. They believe that bacteria could be used to detect and treat various types of cancer, as well as other diseases related to genetic mutations. For example, bacteria could be engineered to identify mutations that cause genetic disorders, as well as to produce compounds that could be used as pharmaceuticals. Additionally, researchers believe that this approach could be adapted for use in other areas of medicine. For instance, bacteria could be used to detect infections or other diseases related to specific genetic mutations. They could also be employed for the delivery of drugs or other substances into the body. Overall, the research suggests that the use of artificially created bacteria could be a promising approach to the detection and treatment of cancer. However, as researchers point out, much work still needs to be done before this method can be applied in clinical practice. Additional studies are necessary to develop and optimize the system, as well as to assess its safety and efficacy.